Okay, let's make a start. Good afternoon and a very warm welcome to you all from wherever you hail in the world. Today's workshop is the first of a three-part series co-organized by EATRIS, European Infrastructure for Translational Medicine, the Radboudumc University Medical Center in the Netherlands, the French Foundation for Rare Diseases, Fondation Maladies Rares, Fondazionete Telethon in Italy, and all under the cover of the European Joint Program in Rare Diseases. A very warm welcome to you all. Today, we're going to have a 2-hour, hopefully very interactive session. So have your questions and your input ready for us, please, to really go over drug repurposing for rare diseases in general, the process, both from a technical and a patient perspective, and to have just a discussion on the main issues and challenges around it. And then we're going to have a second and third sessions later in the month. I'll come back to it in a moment. But first, I would like to thank very much our organizing team. I see the cameras are on here. So we have Rebecca, Agustin, Anita, and Courtney, and Lauren, who have very kindly been supporting this process. Thank you all very much. So Rebecca, who's going to be co-hosting with me today, we have a poll question for everybody to answer. Is that right?
Yes, we can do the poll now, or in, let's say, 3 slides later. You know, once
3 slides later, right? See, that shows how badly I know the slides. So maybe you could put the next slide for me. Thank you. So the workshop series is today's session until 5 o'clock, and then next week, at the same time, at the same day next week, we talk about really about the path towards the market as well. So then we talk a lot about the regulatory, the intellectual property issues around it, and the challenges that you have in perhaps accessing the drug. There are many different types of repurposing strategy. Understanding which one you're on and who are the people that are going to try to stop you is a major part of the challenge. So that we'll talk about that more next week, and then the following week. We'll talk about the more implementation issues around, okay, we've shown that this drug actually works great in this new indication, but why is nobody reimbursing me? Why does nobody want to manufacture this, et cetera, et cetera? Many challenges remain, even when we've shown the scientific value. So without any further ado, we welcome the expert speakers here for today. We're going to have Phil Gribbon, who I'll introduce in a moment, speak about the technical pipeline. And Phil has many years experience in the drug development field, so we look forward to hearing his view on the value chain. And then after that, we'll have some quick questions, and then we're going to pass over to Rick Thompson of Findacure in the UK, who has years of experience in helping patient organizations representing rare diseases find the resources, expertise, and the wherewithal to take a repurposed medicine into implementation. And then we'll have a brief overview of who you need to talk to about repurposing if you have a question, and then we'll have some question and answer. So we'd love to hear from yourselves. What are the challenges you face? And we can see if we can answer those or at least stimulate some interesting discussion. So feel free to put questions in the chat. There's a Q&A button or chat button. Click on that if you have questions, and then in the Q&A sessions, we will select the ones. Next one, yeah. Just a note, due to GDPR, those letters that we all love so much, this is being recorded, but we will also make this available publicly in the future. So there will be an email sent to you later on with a link to the recording online so that you can also distribute it in your communities. Please do so. We'd love for other people to be able to see this as well. Once you've submitted a question or you see somebody else's question you like, you can click on a button to upvote the question. So the more people like a question, the more obvious it is to us that that's a good question to ask. So please be interactive in that way as well. So I think we can get started. Rebecca, what's the next step?
The next step now is indeed the poll, because we want to learn about you. Give me a second to launch the poll. It's about who are you, whether you're a researcher from academia, from industry, a patient or a patient representative, a regulator or other. Okay, I see a lot of answers coming in already. Yeah, very good.
Interesting to see, predominantly researchers, but nice to see that there are lots of others represented.
Still answers coming in. I give it another few seconds.
Good.
Okay, it's getting stable here, so I'll close the poll.
Very good. So,
I will save the result.
Thank you.
So and then share them with you so that we can get an idea. So indeed, big majority of researchers from academia, but also 11 from industry, 5% patients, and then quite a few others.
interesting to have the regulator. We will be asking you questions. Hopefully you'll identify yourself. And very nice that we have a good cross-representation here. So it'll hopefully also be nice stimulating crosstalk. So do please chime up from each of these sectors later on for some discussion. Thanks, Rebecca. Now, I believe we also collected some information about the participants, right? So this is what you filled in when you answered the registration. Thanks for doing that. So what we noticed is that there's a lot of people have really just have heard about it or just starting on this journey in repurposing. So there's a lot of unknowns. So I think that this workshop series is certainly very timely. But what's great is we also have some real experts who are going to be in this meeting, and we'd love to hear from you because we're not pretending to be the experts. We have experts from specific parts of the field, but there is no real discipline called drug repurposer, so we need to hear from everybody. Do please share with us. Nice to see a lot of people are in the process. 65 of the respondents, which I think was about 40% of the respondents, are actually in the process, which is very nice to see. Next slide, please, Rebecca. Also, nice broad spectrum. Italy and Netherlands, no surprise, because we had big pushes there with the co-organizers, but very nice to see. We also have even people coming from across the water, the Atlantic. So we collaborate with Oswaldo Cruz Foundation in Rio in Brazil. They're a fantastic collaborator in a drug repurposing initiative that we formed with them and others called Newfound, together with NIH NCATS. That's why we have also a lot of US representatives. So it's very nice to see that there are people from all over the place. We'd love to hear if there are regional differences as well in your experiences. So thanks. Next slide, please, Rebecca. So without further ado, we're going to now move into the main content of today. So we're going to hear from Dr. Phil Gribbon, who is basically a very well-experienced drug development expert who's now he's director of discovery research at Fraunhofer Institute in Hamburg, but has long experience in the drug discovery process around drug screening. More recently as the coordinator of the EU Open Screen as it was becoming a permanent infrastructure, driving that process very diligently. And then before that, European Screening Port, and before that in the pharmaceutical industry for many years. So Phil, we really look forward to hearing about the pipeline overview from you. Thanks very much for your time. So if you start sharing your slides and take it away, please go ahead. Thank you.
Thanks a lot, Anton. Can you hear me okay?
Yes, we hear you fine.
Very good. Okay. Okay, hello everybody. As Anton said, my name is Philip Gribbon from the Fraunhofer Institute of Translational Medicine and Pharmacology in Hamburg. And I'd first of all like to thank the organizers for giving me the chance to talk about some of these topics today. I'm going to try to present an overview of the application of drug repurposing to the discovery of new therapeutic treatments in rare diseases. As a bit of background, the Fraunhofer has been involved in a number of repurposing efforts in the recent past, particularly in infectious diseases, and we have some track record on the COVID side, and I might show some of that data later on because it's the most interesting and newly generated data, but also in CNS indications. And most recently, we've become part of 2 recently initiated European joint programs in rare disease projects that treat KCNQ2 and the QMLs project. I'm not going to be talking about some of those directly today because they're still at quite an early stage, but some of the methodological approaches that I'll be going over are exactly what we're planning to do within the context of those projects. So I just wanted to start off with some of the sort of challenges associated with the regular drug discovery and development processes in rare diseases, regardless of the modality, whether it's a new chemical entity, a new biological entity, or a repurposed compound, or a gene therapy. Many of the audience are going to be aware of these points, but I think it's just worth revisiting. I'll just get a pointer. Yeah, it's just worth revisiting. So although there are over 350 million individuals who are affected by rare disease globally, the current situation is that over 95% of the 7,000 or so diseases do not have a licensed treatment. And the treatments themselves are in many cases treatments towards the symptoms or comfort treatments, which address second order complications instead of the underlying diseases. And current treatments are therefore aimed at improvement of life quality of those affected, and it doesn't prevent, in some cases, the inevitable decline in function. As this audience will be aware, there are some limited incentives for commercial organizations to discover and develop treatments in rare diseases. And taking into account the process for discovering, developing a novel drug, this is a typically very laborious, costly, and failure-prone enterprise. And I think it's especially unappealing to do for rare diseases that only affect a small number of individuals and therefore generate a reduced profit. And that's the perspective of, let's say, the commercially focused organizations. When large manufacturers and the pharmaceutical companies do invest in research and development in rare diseases, market prices can be especially high and cost of treatment can be high, resulting in higher profits, but potentially reduced access for patients. The development of new drugs in rare disease is a major challenge, and often there's a limited amount of knowledge with respect to the disease epidemiology, manifestations of the disease, the degree of heterogeneity in the disease as the natural course and progression of the disease. And this is reflected in the limited availability of biomarkers for assessing clinical improvements, and also animal models, which are suitable for efficacy testing and evaluating during preclinical stages. So, all in all, the identification, certainly from a new chemical entity or a new biological entity, not a repurposed compound, is an expensive and costly process. So this is a sort of a fairly complicated slide on the right-hand side, but it's a representative of the journey involved in the early discovery of a novel compound up to clinical stages using target-based approaches. And in a target-based approach, the amenability of the project is very much focused on the so-called drugability of the biological target, such as a protein. And drugability is a term that's used in drug discovery to describe a biological target, such a protein that is known to or is predicted to bind with high affinity to a drug. So the binding of the drug to the target of interest needs to alter the function of the target and result in a therapeutic benefit to the patient. So the concept of drugability is often restricted to small molecules, but it's also been extended to cover biologics, medicinal products, and therapeutic monoclonal antibodies. And they are publicly available databases which will allow you to evaluate the drugability of your molecular target of interest. They're provided by Kemble, for instance, which has this drugability portal. But the current situation is that only 2% of human proteins are known to interact with currently approved drugs. And furthermore, it's estimated that only about 10 to 15% of human proteins are disease modifying, whilst only 10 to 15% are druggable. And there is no correlation between a disease modifying and a druggable protein, which really means that only sort of 1.2 to 2.5% of disease modifying proteins are likely to be druggable. Therefore, the number of new undiscovered drug targets is likely to be very limited. So if a target is druggable, then an appropriate assay needs to be established, developed, which is representative of the role of the target in the disease. And then you need to go through a process where you screen relevant compound libraries in order to identify hit compounds. So in the case of looking to identify a new chemical entity, that could be hundreds of thousands of compounds. In the case of repurposed compounds, it's typically less than 10,000 compounds which are within the repurposing collections. And finally, there's a need to perform appropriate safety, there's a need to establish the SAR relationship, so that's a structure activity relationship, and an understanding of the absorption, distribution, metabolism, elimination, and the toxicity properties of the compounds. Although this is for NCEs and not necessarily the case for repurposing compounds. Finally, there's a need to perform predictive safety and pharmacology studies in vitro and in vivo, because it's essential to have an understanding of the engagement of the drug with the target, that the original mechanism of action is relevant. And there's a variety of methodologies for using that, most recently examples such as the cellular thermal shift assay for looking at target engagement. So this is a long and complicated procedure for the development of new chemical entities, new biological entities. And what is this sort of classical timeline? So here's a typical timeline for the development of a new medicine. In this case, it's for inhibitors of PCSK9, which is a hepatic protease that attaches and internalizes LDL receptors into lysosomes, therefore promoting their destruction, leading to a lowering of LDL levels by about 50 to 60% above that which can be achieved by statin therapy alone. So after the initial observation that PCSK9 could regulate LDL receptor degradation, it would potentially be a target for modulating LDL receptor levels. There was an intensive period of hit discovery and screening within industry, which resulted in the discovery of a number of small molecule ligands, which were then also confirmed by structural biological approaches. Unfortunately, there were also issues around these compounds, and the companies involved changed track and they moved into a monoclonal antibody approach. And in the end, the sort of upshot is that in these classical methodologies, whether you using small molecules and then in this case as a backup biological, it took over 12 years from the initial observation of the role of the target to an actual compound reaching the market. So that's let's say the normal non-repurposed situation. So if we look across for all new chemical entities, there's the situations we have low success and a relatively high expense. Again, looking for the general situation, not just for rare diseases, then the cost for bringing NCEs and NBEs to the market estimated to be in the region of between $1 and $1.7 billion. And the overall success rate between phase 1 to approval is about 3.4% for oncology indications and rising to about 32% for ophthalmology. So the overall success rate improves up to 75% for phase 3 studies reaching approval. So the trite message or the easy message here will be that if you start later in the process, then you're going to have a higher chance of success. So you start with a phase 3 candidate. Obviously, it's not as simple as this, and this is where the advantages of repurposing approaches may lie. For example, many of the safety aspects of the compounds may well have been understood as part of the work around done in bringing the molecule to market for the original indication. And as I guess many of the people on this this call will know, that there are a number of incentives for the developments of orphan drugs, which rare diseases is a subset of these. Again, these are for NCEs, could be biologicals, gene therapies, or repurposing, and they all have these incentives to exist, developed by the regulatory authorities and the government in order to promote the extent of discovery here. So these range from extended market exclusivity through to waiving of fees. So since the introduction of these incentives, there's been some success in the field with over 360 drugs approved by the FDA since 1983 for the treatment in rare diseases. And in Europe, we have about 112 drugs, or a bit more than that, have been approved. But the focus here has been very much on rare oncology diseases, although they're at present, they're around 450 drugs which are in the development stages. I think it's also the case for new chemical entities and new biological entities that in order to recoup the costs associated with developing the drugs for these smaller patient groups, then the eventual cost of treatment can be very high. And this can lead to situations where you have very expensive drugs such as Spinraza for spinal muscular atrophy. And this is also linked to the willingness of funders to pay and ultimately patient access. I think finally, in terms of orphan drug clinical trials in general, there is some evidence that phase 1 and phase 2 success rates are higher, but this may well be down to having a more well-defined patient population, which may aid in the prosecution of the clinical trial. So now I'll try and introduce compound repurposing, or also known as retasking or repositioning, where the aim is to find a new use for an existing drug. In the small molecule approach, which I'll mainly focus on today, there are approximately 3,000 or so marketed drugs on which to draw upon, plus a further 2,000 to 5,000, depending on where you get statistics from, compounds which have previously reached clinical trial stage. Some of this latter group also include shelved compounds from projects within pharmaceutical companies which are stopped for a variety of reasons, including safety issues, lack of efficacy, or maybe even commercial reasons. Maybe even a better competitor compound came to the market before that compound did, and it was shelved. So there exist a number of potential benefits with compound repurposing based upon the availability of appropriate safety and ADME data coming from the original discovery and phase 1 studies in the original indication of interest. And these advantages and the availability of this previous data can lead to an overall reduction in the time it takes to and the time and effort to get to market. For example, there's a shorter discovery process where the requirement is to show compound efficacy in a relevant disease model, but not necessarily to perform the complex and expensive medicinal chemistry optimization processes that you would need for a new chemical entity. Similarly, there may be an approved manufacturing process in place for the compound, and that can speed up the availability of material for clinical trials and speed up the clinical trial prosecution process. So overall, the estimates are that for compound repurposing, there's a significant cost and time saving which may be achievable. So the figure here is 2.5 billion for a new chemical entity. For a regular orphan drug or a new biological entity, if we use a compound repurposing approach, the time may reduce, but also the costs may reduce down to about 300 million, and with a potentially higher success rate. However, it's not all plain sailing, and issues still exist. There's a potential for the risk to outweigh the benefits in the clinical trial due to unforeseen adverse events in the new patient group. And there's also potential legal, regulatory, and commercial aspects which are specific to repurposing, which may be important relative to a new biological entity. So there's some very important factors which are not just associated with compound repurposing, but also for all other approaches, whether it's new chemical entities or new biological entities. So before we go into more detail, it's worth looking at some of these factors that determine the success of a drug to pass through the development phase. In this case, it's regardless of the source of the drug. We need to essentially show that we have a clear understanding of the drug, dose concentration, and effect relationship. This means that we have to know that the compound is present at the site of action at the sufficient concentration and time in order to ensure that the drug has an effect in the disease-affected tissue. We need to make sure that the drug is bound to the target, and that the mechanism of action of the drug leads to a functional modulation of the target and to a pharmacological effect in the patient. And this is the case for any disease, doesn't matter whether it's rare disease or repurposing or whatever. But I think one additional consideration that we may need to take into effect in the rare disease case is that different genetic subtypes can exist for the same target. And this means that the same target these can lead to differences in disease and disease progression. So that specific compounds may then only be effective for a subset of patients. And we're seeing the consequence of this in the case of cystic fibrosis, where mutation-specific therapies are being developed. So there's a number of challenges which are associated with compound repurposing in rare disease. We're going to look at some of these now and see how we can address them. I think firstly, and arguably the most important challenge, is the need to identify the right marketed drug which is relevant for our target and associated disease. And here are some of the methodologies for doing that. I think that the second point is that we need a mechanistic assessment of the drug effect in disease-relevant preclinical models. So these are the in vivo studies you need to have access to good models or good translational systems for testing the drug. And then we need to evaluate the efficacy in phase 2 trials based upon the assumption that the phase 1 data is sufficient. And the work undertaken as part of the original indication is sufficient to support it in this repurposing effort. So there's two essential types of approaches when it comes to the identification of the candidate molecule. What you can boil down to experimental approaches, which are in yellow here. So developing biochemical assays to identify ligands which interact with the target, and there's a number of methodologies, and I'll go into more of that later on. And then the use of phenotypic screening methods. Here we're using disease-relevant models. We may be trying to influence a whole pathway rather than just an individual target. And in these cases, a number of new technologies have come on board recently that will help facilitate this phenotypic screening approach, including advanced high-content imaging and the use of gene editing techniques to generate disease-relevant cell lines. But there's also, I think there's this set of, let's call them informatics informed approaches. And this includes signature matching, where we try to compare the signature of a drug in a tissue, such as its transcriptomic or its proteomic profile, with that associated with the disease and the healthy phenotype. We can also identify ligands for the target of interest using in silico screening methodologies, and I'll go into a little bit more of that later on. And we can also use genetic information and pathway information in order to identify relevant targets. And these may be upstream or downstream of the main target of interest, but where modulation of this additional target could lead to the desired therapeutic effects. And finally, we can use retrospective analysis of patient populations that can inform the selection of compounds. And these can be extended into the vast amount of data which is present within electronic health records in order to identify potential treatments there. So I'll try and cover a couple of these in more detail. So first of all, using signature matching as an identification of candidate molecules. And in the signature matching approach, we can analyze the transcriptic, proteomic, or chemical profile or signature associated with drug treatment. And this is called the drug response signature. And this can then be compared with the same profiles generated in disease tissues in order to generate the disease signature. And the principle of signature reversion is that a compound which can reverse the disease signature towards the healthy signature has the potential, therefore, to reverse the disease. And this method is the basis of multiple investigations at the moment, and there's an expanding literature and set of experiences here. And I think this is very much a hot topic at the moment in this area, using these reverse genetics methods. A little bit more complicated or involved is the so-called connectivity mapping approach, which was initiated by the Broad Institute. And this is something which can be achieved using online informatics tools, at least in the first instance. And you'll see a link to some of those resources there. They've made available a huge amount of online data, along with which can be such as gene expression profiles or network-based cellular signatures for tens of thousands of compounds, including a number of bioactive compounds across hundreds of cell lines. And these online tools can be used along with your own proprietary data in order to identify novel connections between targets and diseases, as well as looking to avoid safety issues. So another methodology that we can use is to exploit information that we have on target protein structures to predict the binding between the target and the ligand. So your rare disease target and your ligand in this case would be your marketed drug. This is a very commonly used method, but normally within the workflows of pharmaceutical companies, they're using millions of virtual compounds and doing in silico docking using those, rather than using a subset of the chemical space, which is represented by marketed drugs. I think with the improvements in computing power, this is an easily scalable methodology, and there have been examples of success here, for instance, in the identification of ligands for the VEGF receptor, and this was later shown to be in this study later shown to be active in vivo models. I think the difficulty with the computational molecular docking approaches, or a pure in silico approach, really is linked to the limitations associated with the experimental data sets. They may not be available, and there's whole classes of targets, for instance, with ion channels, where there's only a subset of targets are available, or there may be a considerable amount of uncertainty in the link between the target function and the disease state itself. Now, the first problem about the availability of data may be addressable soon with resources such as AlphaFold from Google, which is coming online, and from our experience here in the Fraunhofer, that seems to be pretty effective in predicting protein structures. And I expect that's going to be extremely useful in the rare disease field in the future, especially for predicting protein structures associated with particular genetic mutations. However, this translational aspect, going from a ligand binding through to a change in the molecular function, through to a change in cellular, tissue, and disease phenotype, I think that's still a considerable challenge, and that translatability isn't always in place. So you can do a lot of work up front to identify ligands, but you might not be able to find anything that's effective using this methodology. Something that has been extremely effective in the repurposing world, and in some cases within the rare disease world, is the use of electronic health records and the use of retrospective clinical information. And there's multiple examples of successful repurposing in the past, and the most high profile is, for example, would be sildenafil and raloxifene in breast cancer. However, many of the instances in which looking at patient information, post-marketing surveillance data, or clinical trial data, many of those were done on a serendipity basis, but not so much on a systematic basis. And it's certainly the case that electronic health records have a vast amount of content these days, and that comes down to images, genetic sequencing, in vitro diagnostic information, etc. But we still have issues around the fact that a lot of the data is not structured, or employs or doesn't employ an appropriate standard for allowing for wide-scale analysis or interoperability between different data resources. And similarly, access to retrospective clinical data may well be a problem in order to support systematic analysis. But it's certainly the case that almost all of or the vast majority of the examples of repurposing have come from these types of retrospective clinical analysis in the past, not necessarily in the rare disease field. But there is an increasing use of electronic health records and the use of large-scale analysis of these records. And this is an example from Pike et al. In 2015, where they were able to get hold of the records from over 500,000 patients. And there was a huge amount of data in there that included over 9 million laboratory tests, as well as genomic signatures, and they put them through advanced bioinformatics and analysis pipelines in order to identify potential drug target interactions and propose, in this case, they were able to propose terbitulene sulfate for ALS. And this is a selective beta-2 adrenergic receptor against agonist, and it has a role in inhibiting protein degradation, stimulating protein synthesis, and reducing neurotrophic factors. There was a positive readout in ALS for this compound in animal studies of neuromuscular degeneration. However, there hasn't been any evidence so far in clinical trials in ALS of this approach working, at least in this area. But it's certainly a fruitful area for the future, especially if we can put in place processes to access these data in a more consistent way to allow for these types of analysis of electronic health records.
For the quick information, you have 5 more minutes. Thanks.
Okay, thanks. So So I'll just finish off with some of what we've been doing with bioassay approaches. For screening, screening-based approaches to identify compounds, it's a prerequisite to have a high-quality compound collection, and the one that we use within the Fraunhofer is based upon a set originally developed by the Broad Institute. And this contains over 5,500 compounds, 3,000 marketed drugs, and a high number of bioactives and reference compounds. We've got examples internally where we've used these repurposing compounds in target-based biochemical screens. Here's a typical example of a FRET-based assay in a 384-well format. And in this screen, we screened over 9,000 compounds and got a number of hits. But then the important point is that you need to be able to analyze those in counter and selectivity screens and also confirm their activity in cell-based assays. And here's an example from those screens where we did identify a number of compounds, and we were able to confirm their mechanism of action in binding to this protease target. And at this point, unfortunately, we're able to see that there's little correlation between the biochemical potency and the efficacy of these compounds in a functional cell-based assay. And this shows the translating from biochemical to a cellular setting. As I mentioned previously, you can also use structural methods, and here's another structural-based repurposing screen that we ran in collaboration with colleagues in DAISY. We did it in this summarizing a huge amount of work here, all the way from protein production, crystallization, harvesting, etc., through to structural refinement and ligand identification. Here is a representation of the compounds that we identified here, bound to, in this case, it's a protease. The compounds were bound to the catalytic domain, but we also found compounds which were bound to allosteric sites. And this shows the power of structure-based approaches in that you can identify the mechanism of action of binding of your compounds, and that can help with engendering selectivity of the compounds. And we were able to follow up these compounds and understand the mechanism of action of binding and discriminate between the different types of compounds. And we were also able to show compounds were active in subsequent in vitro assays. So also in terms of phenotypic assays, we can use these to identify compounds in disease-relevant models. The highest degree of disease relevance comes from the use of primary tissues or IPS lines derived from patients, or which have had the mutation of interest introduced using gene editing methods. We can see here a wide variety of tissue types which can be generated to support phenotypic-based screening for identification of repurposed compounds. But it's important here point out. So we can't see unfortunately we can't see the movie here, but it's important here to see that the endpoint in phenotypic assays is very important in the design of the relevant phenotypic assays. So here we've got inhibitors for an ion channel, and the effects of this compound are quite different depending on the readout. So you see no effect with cell viability. In this functional beating readout, you see a nice dose response, and you can see a calcium flux. In the calcium flux, you see a similar dose response but shifted to the right. So the readout is important, and you need a sensitive functional readout which is hopefully well correlated to your disease of interest. So finally, just a couple of points. Failure does happen, and there's a number of barriers to drug repurposing in rare diseases. And one of the important points is that the original compounds were often identified to have activity against a high degree and a high affinity for their primary target and their primary indication. But this is not necessarily the case in the secondary indication, and you may need to have a significant amount of material to be present there in order to see the effect, and then you hit solubility and other issues there. There still can be failures at phase 2 and phase 3 associated with emerging safety risks in new patients, for instance, in a pediatric versus an adult population. And obviously there's issues potentially around you can get use patents in a repurposing approach, but enforcement of those patents can be those patents can be difficult, especially with generics, but formulation or dose improvements can help things. And obviously there's different regulatory considerations, and we think we'll find out more about that in some of the later talks, and also some of the organizational hurdles in terms of access to funding and access to expertise. So just to finish off, some recommendations for future success and an overall framework for drug repurposing in rare diseases. I think we need integrative platforms that allow data access and analysis, especially for things like electronic health records, and funding initiatives seem to be moving in this direction. But it's also important that there's a standardization in the way that data is represented in terms of ontologies and standards. There needs to be better access to pharma industry derived lead and clinical stage compounds, and that's something that is happening very nicely over in the US with NCATs and not so much here in Europe. There also needs to be better access to the industry phase 2 to phase 4 clinical trial data. Some advanced data mining methods might be able to find new hypotheses or draw new hypotheses on repurposed targets from those data. It's important that there's a systematic investigation of the safety and liabilities of repurposed drugs in the new patient population in order to avoid issues there. And there needs to be improvements in the way that investment is incentivized in terms of exclusivity, regulatory, and barriers. So here's just an overview of what a discovery strategy might be. And I think we'll put a link into some of the papers that I drew upon for the presentation today, and we'll put them in the chat. So thanks a lot, and I'll pass back over to Anton.
Outstanding, Phil. Thank you. That was a phenomenally comprehensive and strong overview of both the traditional and then vis-a-vis traditional the drug repurposing process. Many thanks for that, for taking the time to run us through it. We have time for a couple of questions here, and then later on we'll go into a bit more in depth. So I've seen 4 come in, upvoted. Let's see which one's most popular. From Peter Bram at Hun. When comparing a drug repurposing strategy versus conventional, is there less need for animal model validation studies? So reduction in animals because the safety toxicity data is there, essentially.
Yes, so I think in terms of the efficacy part of those in vivo studies, then I think the need will be similar regardless of the origin of the compound. I think, but as mentioned, if there is good toxicity data, but in dog models and other sort of reference models required by the regulatory authorities, then there may be an opportunity to that you may not have to do a fully comprehensive studies there. I think you have to engage with the regulatory authorities in order to confirm that. But certainly, there may well be an issue though if you're proposing to change the formulation, if you're proposing to change the dose, if you're proposing to change the dosing regime or the length of time of exposure. And in those cases, it may be necessary to redo the toxicity studies if dramatic changes to the way that the drug is presented to the intended patient population happen.
Yeah, thanks a lot. Yeah, so basically it is very much case dependent, isn't it? But indeed, sometimes the regulator, and this is a related question that from Joanna NSNS. Let's say you're repurposing based on this serendipitous finding in the clinical environment, and you're able to show on under controlled conditions really good results, and you show really compelling data in phase 3. You think the EMA or the FDA is going to approve it without actually knowing the mechanism of action?
So my understanding is that a mechanism of action is not a prerequisite for a successful clinical trial approval or successful drug approval. But obviously, it's extremely useful to have it, and it can help with all sorts of understanding all sorts of potential liabilities of the compound. It helps you when it comes to understanding there may be some variation in the distribution of your target of interest in different tissues, and again that links through to liabilities, PK, PD, etc. So I think it's not a prerequisite, but it's certainly extremely useful and will not harm your case at all if you have a very solid understanding of the mechanism of action of your compounds. And there's multiple drugs on the market where the mechanism of action is not well understood and it's still under investigation. So that those examples exist, but I think in our case, or in the case of rare diseases, many of them are monogenic in origin, and you do need to, I think, demonstrate that your compound or your therapeutic agent is modulating or is interacting with your target of interest, or it could be interacting with upstream or downstream components if that target sits in some sort of signaling pathway, but whereby you get the same effect. But I think having an understanding of that is really very important. But if it works, then that's okay as well, I guess. Ultimately, the pragmatic approach.
Understand, yeah. Indeed, where the problem might arise might be getting the authorization for the phase 3 if you aren't able to show a reasonably credible rationale for why you think this is going to work. And I think that's where you may get into a bit of trouble indeed. I do invite regulatory experts on the call to also use the chat. Please chime in. We're not going to open the mics yet, but in the discussion session later. Phil, that was really fantastic. Thank you. We'll leave the questions there. And
I think we could do one more question, Anton, if you like.
Great. Yeah, that'd be great. Thank you.
I think the final question is quite good about the 3D
sources for signature match.
No, the 3D screenable assays.
Yeah, good.
Yeah. So I think that's something that we are attempting to do. So that's why I've highlighted that. I think some of these 3D systems require an enormous amount of work in order to get them to the point where they're representative of the disease tissue of interest. So your cell culture might take several months, for instance, if you're making a mini brain. But the data that I've seen and the information that I've seen is that you're screening a lower number of compounds. You're not screening millions of compounds. You want to have the richest data source that you possibly can. And if you can demonstrate that your 3D microtissue, mini brain, mini liver, mini heart, whatever model is really the closest that you can get to represent the underlying disease state, then I would say definitely use those and exploit those to the maximum. And I think repurposing maybe if you can get them to the kind of scale of running in the hundreds to the thousands, then I think you have a really good opportunity to identify very interesting compounds that won't emerge from a kind of classic structure-based biochemical screen, 2D in vitro screen approach. I think there's a huge amount of effort there. I think the difficulty with the 3D systems is what the endpoint is. So it's very difficult to get, let's say, conventional reproducible endpoints using classical imaging-based methods and other methods. So that's, if you can get a good endpoint, a good biological system and a good assay system, then I think it's the way to go.
You're mute, Anton.
Thanks. Sorry. Understanding, a big part of the challenge is also understanding what the assay can and cannot recapitulate, or really where is it valid and where not. And I guess that you have to really work closely with the experts to understand the limitations.
Yeah, and reference compounds are always useful there, but oftentimes we don't have them, and that's part of the issue as well.
Yeah, super. That was outstanding. So thanks so much for the input. I'm sure that we're going to make contact details for the various contributors today available for people who might have specific questions. We'll come back on that later on to make sure that people are contactable thereafter. So I'm now going to introduce Rick Thompson, based in the UK, who is the CEO of Findacure. Since 2017, Phil, Rick has been active there. Apologies. Thanks. I'm looking for the slide from Rebecca for the coverage. So Rick has a PhD from Cambridge University and has been the CEO for Findacure since 2017, who have been helping patients and patient groups in rare diseases do repurposing strategies and really help get to the clinic and beyond for these very serious diseases. So we look very much to hear about the patient's perspective from Rick to really show us the examples of how it's done from that side, from the pool side. Rick, please go ahead. We look forward to your time.
Thank you so much, Anton. Hear me okay? All good.
Perfectly.
And hopefully you can see my screen as well.
Perfectly.
Perfect. Brilliant. Guys, it's a real pleasure to be here today. Thank you so much for inviting me. And I really enjoyed Phil's presentation too. I've given many talks similar down the past, but never quite so with such detail or authority. So Phil, really enjoyed that. Thank you so much. I, as Anton said, run a charity. So my perspective is somewhat different and coming from a different background to you guys, but I've had a background in research myself and really keen to drill into how the patient community in the rare disease space are trying to use repurposing strategy to move things forward. And in the work that we do at Findacure, we spend a lot of time thinking about that. So I'm going to be talking from the perspective of someone who's observed lots of work happening in the space and the trends and patterns that I'm seeing in that repurposing world. The final thing to mention is I think there's a few things in the early slides where I'm kind of copying and going over some of the ground that Phil's touched on. I'm going to try and keep that a bit lighter in response to that, because I think Phil gave a really excellent introduction to the setting. But I will touch on a few facts and tweak how they are relevant to that patient perspective in that world of repurposing. So to do that, we want to set the scene, and this is an infographic we use at our organisation to highlight those key rare disease statistics that hopefully we're all really very familiar with. Rare diseases are obviously individually rare. EU definition is a condition that affects fewer than 1 in 2,000 people in the general population. But that does scale up to be quite a large and significant population at a global or European or even national level. About 35 million in the EU is what Phil stated, and a number that lots of people share. And it's really that collectivity between all different rare conditions that begin to make you see the size and scale of the problem that are faced. Obviously, the genetic background means that a huge proportion of those affecting children, and there's a really high infant mortality across the different conditions, with about 30% of rare disease patients estimated to pass away before their 5th birthday, which kind of highlights the severity of many of these conditions themselves. Now, obviously, they're very disparate. We've got 7,000 different conditions. They affect all different systems in the body. There can be very different prognosis and very different impact on life. But what we're seeing in rare diseases is actually commonality in the experience of having and being diagnosed with a rare condition. There's a commonality in a prolonged diagnostic odyssey, taking multiple years, multiple misdiagnoses, multiple doctors to actually finally figure out what is going wrong with you as a rare disease patient. And when you can get to that diagnosis, generally there's a complete lack of knowledge, understanding and information about how to manage that disease, let alone hope of treatment. Patients with rare conditions can struggle to hold down paid employment, they can struggle to access effective education, and obviously struggle to access care. And this is a really challenging situation which is shared across those different disparate conditions. What we've seen that leading to, though, particularly since the rise of social media, is a patient population that is driving change itself. They are informed, they are active, and are determined to deliver change for their own rare condition. Essentially, these patients are coming together, seeing little research happening in their condition, seeing little hope of treatment development, and just seeing it as a hopeless situation, but recognizing that together they can deliver change themselves. And we're seeing the rise of the rare disease patient social entrepreneur, people who are going out there and trying to use basic business skills to drive change in their particular condition area. And a lovely example of this is the CATS Foundation, which is a UK-based patient organisation for Tay-Sachs and Sandhoff disease. So as recently as 2010, there was no patient organisation whatsoever for Tay-Sachs in the UK, and very little research going on across Europe. In 2011, though, 2 parents, Dan and Patricia Louis, came together to found CATS, Cure and Action for Tay-Sachs, after the diagnosis of their daughter Amelie with Tay-Sachs. And their initial aim was simply to find other people with that experience, understand what they could hope for their daughter when they were essentially giving themselves a Google printout from the doctor. They formed an organisation which provided support for others and began to really think about research, running patient conferences and then bringing in researchers into those conferences to drive forward the research agenda. Through doing this, they've been able to get centrally involved in a gene therapy project, which is moving forward in Cambridge here in the UK, but also a range of other different research projects, including a drug repurposing trial, which is ongoing in the UK. There's now a huge range of different research studies and clinical bits of research and trials going on across Europe, primarily as a consequence of the action of this patient organisation and their determination to build an effective registry that details the information and experience of those patients around Europe, and that they've really transformed the landscape in a matter of 11 or 12 years in Tay-Sachs. Now, unfortunately, and a key thing to mention is that for Dan and Patricia, that is too late. Amelie passed away a few years ago from her condition, and they are still continuing the fight for the families and community they've built around there, but it won't benefit themselves. And this kind of final point really hones the drive of the individuals who are trying to drive this change, and the desire and need for change, and the huge unmet need in the rare disease patient space. Now, my own organisation, Findacure, we try and work with these types of people, these types of groups, to help them drive things forward. Our hope is to see a world in which all rare diseases have treatments that have been made together with patients for patients, and that togetherness, that collaboration, we think is crucial in the rare disease world. If we can get that collaboration between industry, academics and patient groups, we're going to accelerate treatment development, we're going to accelerate new diagnoses, and we're going to accelerate the care that patients can experience, and really address this huge underlying unmet need in the space. So if we're saying there's this huge unmet need, generally you're going to go to these things as the way you're going to tackle rare diseases, right? You're going to think about the exciting potentials of de novo drug discovery. You're going to think about genomics, how it's revolutionized the way we view medicine and rare diseases now, with 80% or so being genomic. You're going to think about the potentials of personalized medicine and tailoring your drug or treatment to the specific mutation that each individual patient is tackling. And of course, you're going to think about the really exciting world of gene editing, where you can potentially change the underlying genetic code within the individual themselves, and that should clearly revolutionize the way we deal with and treat genetic conditions. And all of that is true. All of these are exciting, sexy forms of science that we should be getting excited about. And if that's the case, why bother with something like repurposing? If you like your medicines new and innovative, is repurposing really going to be ticking that box for you? Now, I think it will, and actually I think Phil's already done a great job telling you why, which is fantastic, and I'll touch on a few of those points again. But I particularly think it's crucial to think about this from a patient perspective, because for those patients who are living with these conditions, there's a need for change now, yesterday, not for 15, 20 years down the line, when we might finally be able to see an effective gene editing treatment for conditions. And repurposing has potential to fill that gap in the meantime, until some of these more advanced therapies can come online for what are often ultra rare conditions. Phil's already touched on data like this, so I won't spend long on it, but essentially this graph is showing the key point here, that over the last 10, 20 years, 30 years even, we've seen a reduction in output in terms of the new drugs that have been discovered from industry, and the general increase in the R&D spend to produce that smaller amount of drugs. Overall, meaning the cost of developing a new de novo drug is going up, peaking around here at 2.6 billion US, sitting around at 1.8 in 2013 that Phil mentioned earlier. So this suggests there's a reduction in efficiency in our drug development process. And when you pair that with the unique situation in rare disease, the fact that you have these small patient populations, meaning there's a necessity, it is argued, for very high prices to deliver those drugs. You can see a real challenge in this area. In 2016, for example, you can see that a non-orphan drug was coming out at around $28,000 US per patient per annum, compared to an orphan drug coming in at over $140,000 per patient per annum. In that kind of situation, if we're going to try and drive rare disease treatment development, it's going to be unsustainable for society to fund many hundreds of different treatments at that kind of price point. Because of that, because of the slowing down of de novo drug discovery and the high prices necessitated by de novo drug discovery for rare disease, we feel a new model is needed to try and tackle the huge unmet need in rare diseases, a new model that can work alongside existing routes for drug development. And I think repurposing can be one of those key models to do that, and it's beginning to fill that role. Phil's hinted at the idea that there's many different targets and pools that you can tap into when you're looking to repurpose drugs. That existing pool of generic drugs is a really rich body of pharmaceuticals that we could tap into and make much more efficient use of, particularly for some small ultra rare conditions, if the right data is existing. They're available, they're low cost, and there's a wealth of data on human use, and that makes them very appealing theoretically for repurposing. What the challenges come in there in that space, though, is how we secure intellectual property and therefore develop an effective business model to drive these types of research forward. And we think there's a bit of a market failure in this area that's stopping that working so far, which I'll touch more on later. Alongside that, we've got current available branded compounds, and repositioning those is a great route to extend the license for companies, and we're seeing lots of that happening, as well as those shelved compounds, drugs that have been developed and proved safe in humans but didn't prove efficacious for original development, and now sitting on a pharmaceutical shelf somewhere waiting to be used for something different. And we're seeing increasingly more ways that these have been tapped into and used in rare diseases. And again, what Phil's really hinted at, but is key, I think, in repurposing, is that you're working with something that you know, and that allows you, hopefully, to develop those drugs, whether they are generic, existing, compound, or shelved, in a more quick and a more cheap manner, and that can only be good for developing rare diseases. We remove that long, costly de novo drug discovery step. We know something about the safety profile and side effects of the drug, which can save us time. If it's a generic, we have a lot of history of human use, and that means we can reduce that requirement for some of those early stage clinical trials looking at safety. And we know about the pathways of action, so that when we're really looking for potential candidates, particularly in that small or ultra rare space, we can reduce the number of drugs we're screening from our tens of thousands to thousands or even hundreds to more effectively, quickly find a drug that could have a benefit for your ultra rare patient population. This potential, therefore, for increased speed and reduced cost makes repurposing an absolutely ideal model for a collaborative drug development and collaboration between patient groups and academics together, with some industry input down the line. When we're talking about patient engagement in research, I think it's really crucial to recognise there is a wealth of different ways and areas that patient groups or patients themselves can influence the way we go through that process, whether it's repurposing or not. And they can impact from the preclinical space through trial design, trial itself, out into reimbursement. We can see, obviously, charities having a big impact in funding research and initiating that research. But even in this preclinical space, they can have a much bigger role thinking about the collection of natural history data, as in the case of cats, building that registry, really helping to instigate research down the line, or thinking about the ways they can bring stakeholders together to drive and attack major issues, priorities for the patient community in that rare disease. We're seeing patient groups now increasingly get involved in trial design, or protocol review, or even refining site setup at clinical trials themselves to make sure the trial is more effective and doable, if you like, for their patient community. Groups are also having to support patient recruitment and retention into trials, and then thinking about the ways you interpret the data, which may help with the demonstration of efficacy, but may also help in the process of approval and reimbursement, trying to show what this actual endpoint means practically to the lives of the patients you're trying to support, demonstrate the true value of the treatment, which is becoming increasingly crucial to get the drug approved and to market. Now, an example of this is a condition I've worked with on and off over my time at Findacure, which is Wolfram Syndrome. So Wolfram Syndrome is an ultra-rare genetic condition. It affects only about 60 people in the UK. So I can't remember the prevalence, but it's in the region of 1 million, I should think. It affects young people from around the age of 5 and then progresses from there. And around this age, patients tend to exhibit diabetes, both mellitus and insipidus, along with some visual impairment. And from that stage, we see a gradual degradation in both their sight towards late teens, and then tends to progress in degradation of hearing as well. So we've got people that are deaf-blind in their early 20s. From there, there's also further neurological complications, as it's actually a disease that causes degradation in the brain stem, which can lead to respiratory and breathing issues and becomes ultimately life-limiting. So it's a really unpleasant condition, and one that's very hard to hear the diagnosis of at a young age. Now, there's currently no approved treatment for Wolfram syndrome, but there is an active specialist service here in the UK, based in Birmingham, that manages a number of patients with the condition. And essentially, at its inception, was simply monitoring and helping to provide baseline care for these patients. Alongside the specialist service, there is a patient organisation, Wolfram Syndrome UK, who founded to help support those patients that receive the diagnosis and help them connect and work with one another. But they also, like CATS, decided to run patient conferences and began to invite the researchers to those conferences. And the founder and CEO of this organisation told me that at one conference they decided, it wasn't enough anymore for those clinicians and researchers to be simply monitoring the patient. They wanted to see something different. They wanted to see some change and action that could actually improve the long-term prognosis and future for their patients. So she says that she basically locked a number of clinicians in a room at a conference one day and told them not to come out. They had a plan of what they could do to begin to make a difference, to develop new treatments for the condition. And that sounds like an extreme route, and I hope one or two of them at least were expecting it. But there was something that came out of that enforced meeting. And there was a plan that they put together to think about how they could develop a simple drug screen using existing cell models with knockout mutations, just as Phil has outlined, to identify potential repurposed candidates for Wolfram syndrome. Now, using that route, Professor Tim Barrett, who's pictured here, who runs that specialist service, has worked in collaboration with a number of people to identify the potential of sodium valproate, a commonly used generic drug treating epilepsy normally, as a potential treatment to halt or slow the progression of Wolfram syndrome in those patients. He developed cell models, animal model testing, and then used all of that information that essentially he built with a small research team out of Birmingham to secure MRC funding for the first clinical trial of sodium valproate Wolfram syndrome. And I think the first clinical trial in Wolfram syndrome in the world, actually. There's another now working with a different compound in the US. And this just shows the simple impact that patient collaboration can have to instigate research and drive it forward. But alongside that, this patient group has also been helping to support the trial recruitment, the trial retention, and really helping to ensure that the patient's perspective on how the trial is run and its outcomes are fully understood by the patient, by the clinicians and researchers themselves. And it shows how that very tight interaction can make a difference, even when there isn't a strong scientific basis or scientific officer position within the patient community. Now, we're seeing a lot of this type of interaction happening and a lot of this type of research as well in rare disease space. Really highly ultra-orphan conditions who manage to find a researcher, a clinician, or a small team to try and push things forward and think about how they can collaborate together to make a difference in an otherwise untreated condition. And I think there's lots of research happening in this area. But what's common with all of these type of projects is generally a lack of clarity about what the strategy is to take this project beyond simply demonstrating efficacy and actually to deliver impact to the patient, i.e. to reach market in pharmaceutical speak. The people setting up these projects need to probably spend a little more time considering the strategic decisions they need to make to find that pathway to market. And that will come dependent upon whether they're hoping to prescribe the drug in an on or off-label route, on-label always been preferable, whether the drug is currently generic or under patent, and how then they have to bring in other collaborators from industry to try and move things forward. And many of these groups are trying to move things forward in a really rapid way with little support, and giving them the support and guidance they need structurally to tackle things like securing placebos for trials is really crucial. In the case of Wolfram syndrome, they're working with a generic drug and they're aiming for an on-label route. They've secured EMA advice and recommendations for their protocol and are trying to move that forward there. But it's this type of strategic consideration, which I've kind of placed here for your perusal, which I think is crucial and a big stumbling block for many of these small projects moving forward. We conducted a review of repurposing in collaboration with a consultancy here in Cambridge a while back. It's a kind of an ongoing project that's not been completed at this point, but the main thing I want to pull out from here was that we're seeing a lot of this type of repurposing happening in the space. We identified over 167 different cases of repurposing for rare diseases happening in the literature. And there's two things to pull out from this slide. I think firstly, we can see a lot of these are reported as off-label use or based on case reports. And now I think that's really fascinating, but also I think these are going to be highly underreported types of research that are happening in the space. So I'm sure far more is happening in this kind of clinic-led innovation role. But what we are also seeing in this data set is a very small number of those reported through small clinical trials or retrospective analyses. And this again is suggesting that there's a challenge in moving this kind of clinical innovation into a more formal space. And there's another challenge we need to overcome. How can we find more funding to drive this type of ultra rare clinical trial work into the fully reported clinical trial space? One route to achieve that, obviously, is to move into collaborations with industry. And this is an example of the repurposing of the drug everolimus, which is a specific derivative of rapamycin, a drug which I see used for everything these days in repurposing, into the space of tuberous sclerosis. Now, this project began with pure academic fundamental research into TSC, tuberous sclerosis, trying to identify the underlying mechanism of the condition. What's different here is that the researchers who found that mechanism also recognised there was a pathway for inhibition of that mTOR pathway, and there were lots of drugs there active that could do that. And rather than saying that's a nice conclusion, there's my paper, decided to actually go on and try and move things forward and do the research that could see if they could drive this to treatment. So they decided to try and put together more evidence, built the evidence to show it could have potential, and then put a bid together for running a first-in-human trial, an open-label study. But that was unfortunately rejected because it's using a generic drug by the MRC. So at that point, they thought this is a dead end. There's no route further to testing, which I think is where many studies in that previous slide are getting stuck. In this case, however, they moved to collaborate. They happened to bring in the Tuberous Sclerosis Association in the UK, who were able to provide a chunk of funding to help fund this research and move it forward. And the TSA also were able to bring in funding from the Welsh government in this case, as the research was based in Wales. And what this meant is they were able to run a small open label phase 2 trial to look at the impact of everolimus on kidney tumours and tuberous sclerosis. Now, this open label study was no way going to be enough to lead to a marketing authorization for the drug. What it was enough for was to attract the attention of a large pharmaceutical company, in this case Novartis, who saw the evidence that had been generated, saw a positive outcome from that phase 2 trial, and used that as the basis for them to go and run a fully funded large international phase 3 study of their existing on-label licensed drug everolimus, which was under patent, into tuberous sclerosis. So what they essentially did was access the benefits of orphan drug designation to move their drug, reposition their drug into this market and extend their patent life from 2013 out to 2021 by moving into this whole different area. And now there are multiple different indications of tuberous sclerosis that everolimus is licensed for, and patients are receiving access on a regular basis at a price which in the rare disease world isn't so high, £36,000 per patient per annum. This is a really nice story, again, showing how that initial engagement and focus from both researchers and patient groups can lead to a route to market. The final thing to mention on this, though, is that if that original study had been funded and if there was a good, strong, robust existing pathway to move that type of research forward, the estimated cost of accessing the generic drug could be as low as £250 per patient per annum, which is a vast difference to the cost that health services are now paying to treat those patients. So more foresight, more pathways could have led to much greater savings down the line as well with the same patient outcome. Now, the final big example I want to talk about, which I think is one of the gold standard examples of patient collaboration in drug repurposing, is Alcatenuria. So Alcatenuria is a rare condition otherwise known as black bone disease. And this affects in the region of 1 in a million people. It's an inherited genetic condition and it affects one enzyme within the body. It's a progressive disease, and the enzyme that's affected causes a buildup of homogentic acid. So this acid essentially attacks the cartilages and bones in the body throughout life and physically turns them black, but this actually causes them to gradually degrade over time and lead to a very painful early onset form of osteoarthritis and lots of joint issues for patients as they enter their kind of early middle age, but also can cause calcification of things like the heart valves, which can lead to many further complex complications of the condition. The other thing about Alcatenuria is that it turns your urine black when it contacts with air. So it's a relatively easy condition to diagnose and identify, but this can have implications actually when you're trying to drug the condition and demonstrate impact, which I'll touch on in a moment. Here we can see the pathway for Alcatenuria, the drug work that goes wrong, and you can see basically there's a breakdown in one enzyme here, homogentisate 1,2-deoxygenase, which leads to homogentic acid building up. Now, on this pathway, there happens to be a drug called nitisinone, which is already approved for the treatment of tyrosinemia type 1, which actually leads to death in infants very early without drugging. Now, if you treat patients with Alcatenuria with nitisinone, you completely prevent the formation of homogentic acid. So logic says this should be a very simple, clear way to eliminate the buildup of that problem and therefore eliminate all of the symptoms of AKU. And that is a strong hypothesis, a really easy case for repurposing. So a patient association decided to try and make the case for this and test the repurposing of nitisinone into Alcatenuria. And that group was called the AKU Society, based here in the UK, but they did this out of a failed trial of that very theory in the US. Now that trial in the US failed because of the endpoint selection. What they decided to do was select an endpoint of range of motion of the hip, and trying to see if they could improve range of motion of the hip alone by treating patients with nitisinone. And this kind of highlights the problem when you're developing clinical trials. Nitisinone and AKU affects the whole body, every system, every joint. And when you're going to drug people with nitisinone, you're going to stop them building up the acid, which seems not very likely necessarily to improve the range of motion. So obviously, in this case, the trial unfortunately failed. Despite that, the AKU Society decided to take this on and run their own research program to actually repurpose the drug into Alcatenuria. They funded and conducted the first human autopsy in AKU to get a full understanding of the impact of the condition across the body. They developed the first Alcatenuria mouse model to allow them to really test in animal models the potential of this drug. They developed their own AKU severity score, which tried to assess the disease progress right across the body and to use that as a trial endpoint, which encompass all the different complexities of the condition. They then secured a specialist centre in the UK to treat and manage AKU, and were able to secure the right to prescribe nitisinone to those patients off-label, conducting an open-label study in that setting. But they then went further, recognising the need for a full clinical trial. They applied for FP7 funding from Europe and built an international consortium, which included a pharma company, a clinical research organisation, and patient groups across Europe and clinical sites across Europe, mainly in France, Slovakia, and Liverpool, to run that trial. In doing so, they were able to secure over 6 million euros in funding to run a phase 2 and phase 3 clinical trial to test the efficacy of nitisinone in AKU. They got EMA advice while doing that, and the EMA told them that if they can show there's a significant change in the biomarker, i.e. the levels of homogenetic acid, that could be good enough to get approval. But regardless of that, they decided to push for an endpoint which showed clinical benefit, physical benefit to the patients as well. And despite the rarity of this condition, they ought to recruit over 140 patients onto their phase 3 study, while also excluding most of the English patients who are already getting that drug in Liverpool. Furthermore to this, as I mentioned before, the drug is so effective, it knocks down the level of HGA to almost zero as soon as you take it. So patients knew whether they were on the drug or not in what was a randomized controlled, placebo-controlled trial, because if they were on the drug, they didn't have black urine anymore. And those patients actually retained on that trial. I think they had 5 dropouts across the 3 or 4 year trial period, which shows the value of having that patient support group there involved with the recruitment and retention throughout the study. So they completed this trial and got an exceptionally positive set of data. Not only did they show the halt of progression, they also showed that they improved the outcomes of patients, which was far beyond their expectation. And that drug is now fully approved, licensed by the EMA, and available and reimbursed within the UK and other European territories. So I think this is a great example of what patient groups can do. And I'm going to summarise now, Rebecca, apologies. So this is kind of a summary of where I'm at, and I think a great example of what patients can do in the repurposing world. I think drug repurposing is an ideal collaborative model for patient groups. Academics and industry all have a key role to play in this space. Patient organisations are out there now actively seeking ways to drive research forward, and I'd encourage all of you to find ways to work with them if possible, because they can bring a huge amount of value to the research that we're trying to do. It's really crucial to have that patient engagement to really effectively build a drug development pathway, whether repurposing or not. And it's important also in any project where you're doing this to think about what your endpoint is, what your route to market is, because it's no good getting a nice measure of efficacy if you can't then make it reach patients. And there are clear challenges to how we go about putting this repurposing together, but hopefully there's going to be some changes in the future going to make this much easier, and we'll see a real rise in collaborative repurposing in rare diseases in the future. So sorry for pushing time, but thank you much for your attention, and I look forward to taking some questions.
Wonderful, Rick. That was really, really interesting and incredibly good overview from the patient perspective. Wonderful to see the work that Findacure has been doing with the communities and the very many different communities. You were, I think, perfect on time. We can take a couple of questions, which I think we have here some interesting ones. So from Joyce Nabers, what's your perspective on when the regulators that are involved in the reimbursement decision process, health technology agencies and insurers, when should they be involved to ensure that the data you present them will be fit to task?
Yeah, it's a really good question. I think as soon as physically possible, as soon as you can encourage them to be, is the answer we can always give, and we keep hearing from them as well as any other project. I think in repurposing that's particularly key. A lot of the reimbursement agencies we're speaking to are aware that there are more repurposing projects coming along, but there is a philosophical challenge in their head with paying very large sums for drugs that are otherwise available cheaply. So those bodies would like to be involved early to understand what the benefit is to patients, understand what the route to market is, and to provide advice and ideas on how you can get there effectively. But I think increasingly in rare diseases, we're seeing that regulation is one hurdle and reimbursement is a further hurdle. And I think the sooner you can bring that into any development plan for rare diseases, the more likely you're going to be successful.
Indeed, and I think it's worth pointing out that especially if you're having publicly funded or non-dilutive capital funding for your clinical trials, there are many, many organizations that will happily join in as a partner and help you conduct an HTA analysis, a health technology analysis, help you design the trial so that it's going to have, be able to provide the evidence to show that it has a good positive cost-benefit profile, and could even design a strategy to engage some key regulatory agencies.
Yeah, and I think a lot of regulatory bodies and reimbursement bodies give excellent advice, and are really happy and keen to give advice to people from the academic space as well. And I've had some really good feedback on academics who've been in there and involved in those meetings, and have found it exceptionally useful and open to helping them through the process, which I think is encouraging and we want to see more of.
Yeah, indeed. It's worth bearing in mind that the drug regulatory process, so the authorization of a drug in the central procedure by the European Medicines Agency, is a completely different process to the national process of reimbursement, which is 27 different regulatory processes. However, there has been a lot of work in recent years to try and bring these a bit closer together. And there are instances of actually being able to have a joint meeting with both the EMA and a joint panel of the HTA agencies to really talk about what's necessary to show evidence on both sides. So there is movement in that way, but it's still reasonably complex. I think we'll definitely touch more on the subject in the future as well, in the next 2 sessions. A question from Peter Bram: were patients involved in the definition of the primary outcomes of the Wolfram Syndrome trial? Especially for a disease where there's a wide variety of outcomes, is probably a very important consideration.
Yeah, that's a really good question. Patients have definitely been consulted and discussed with them. Whether they were involved in the final decision on what the primary outcomes were in that study, I couldn't tell you for certain. But I know that Tim has worked very close with patient groups and a few other groups in how they're looking at that. But I think Tim had to take quite a strong steer from the EMA when he was defining what they would do. And ultimately, I believe the primary outcomes come down to neurodegeneration. I think there's actually scans they're doing at the back of the brainstem for that project, which is less patient relevant, but there are specific patient relevant outcomes that are in there. So it's one of those balancing acts, I think, in all of these places, trying to make sure you have the patient relevant outcome in there where possible, but also making sure you're finding the outcome that really meets the regulatory approval and has the robustness that they're going to be seeking.
Clear, thanks. And in your experience, do patient organizations have a preference for whether the drug is generic or it's an investigational shelf compound, or it's a marketed branded drug?
Yeah, that's a tough question. My experience is simply that patients want to see something that gets to them. And I think they don't really care what that route is, as long as it's going to be effective and it's going to actually be accessible. There's definitely increasing frustration over the last few years where we're seeing cases where, whether repurposed or not, there are drugs that have been developed in rare diseases that are effective and efficacious, but the price is so high that patients just can't ever get access to it. And people are effectively moving beyond the age where they can access that drug, or passing away without treatment. And this is leading to lots of frustration and issues between some of the collaborations there, and tensions between either reimbursement bodies or companies and the groups. So from my perspective, I don't think what you start with matters. What matters is you have a clear idea of how you're going to actually get it to the market effectively, and that whenever you're working with patient groups, you try and be very clear about what that plan is and what the ultimate price point will be, and listen to the feedback there about how you get it through, because the evidence of value for reimbursement is king right now, and you won't demonstrate that without patient engagement and involvement as well. If that remotely answers your question.
That's perfect. I think absolutely. And yeah, indeed, whatever works, whatever gets to the patient is probably the best way to answer that. Indeed, thanks, Dirk. There was one more question that came in via the chat, but it was quite an interesting one. In your experience, how and where would you get the funding for a repurposing project that really shows no route to a reasonable commercial return for the venture capitalist or the pharma company?
Yeah, this is a really good and challenging question, eternally, to be honest with you. We've done some work on this down the years, trying to work out new models that can fund generic drug repurposing, and there's clearly a market failure here. There's definitely the willingness to fund from certain government funding bodies or large charities, so organisations such as LifeArc in the UK that have a large philanthropic fund that can drive these things forward. In many cases as well, when a drug is generic, the best route is to find a way to reformulate in some manner, which can then generate industry interest, and that can give you more IP, which will give you that chance to then move it forward in the traditional route. But there's a lack of other models. We were trying to look at a model called a social impact bond, which is trying to develop a socially financed structure which can encourage the repurposing of generic drugs, simply through the fact that down the line, if that drug is approved, it's going to save money to the healthcare payer, because they're going to pay a very low cost for a very significant healthcare improvement in the patient population. So the idea essentially was to get upfront investment to fund the research, and then get the healthcare payers to pay back some money they're saving if those drugs prove to be successful. We think it's a really fascinating idea, and models like this are interesting models, but very hard to encourage large bodies like the NHS in the UK or other healthcare payers to back. But I think these types of models are going to become increasingly prevalent, and hopefully we'll begin to get to the place where those can be more open soon.
Great, thanks. And I suppose it's also further complicated. There can be cases where, for instance, it's a generic that nobody makes anymore. So even if you can prove the evidence, who's going to run the cost of running a GMP manufacturing line for a disease that affects 500 patients, for instance? There are a lot of questions that still have very few implementation answers. There's a lot of policy work that's happening. So there is the Drug Repurposing Observatory run by the EMA that's involving governments and agencies. And we hope to see that over the next few years, there's going to be development of this. And I think in the next couple of sessions, we should be able to also go a bit deeper into these implementation challenges. Rick, I think we're going to leave it there. I see Rebecca's lovely face on camera. Rebecca. Thank you. Sorry to interrupt. Rick, thanks so much. That was really outstanding. And we really appreciate you taking the time to share experiences with us. Much appreciated.
Thanks, Anton. Dear all, we have now the third presentation of today, which will be done by Anton. Anton is the Operations and Finance Director of EATRIS, the European Infrastructure for Translational Medicine. His background actually is in engineering and SME administration, but with the EATRIS, he works now as a specialist in establishing and executing strategic public-private and public-public collaborations, with the real ambition to move biomedical discoveries into patient benefit. And Anton is going to tell you about how to find expertise and resources with respect to repurposing.
Thanks very much, Rebecca. I will share my screen. And while that was my CV, my colleagues just used the short version, which is they call me the bullshit generator. So just bear in mind, caveat emptor, what you hear from me now.
Do you want to share your slides? Oh,
no, it's okay. No, thanks.
Okay.
Please continue. That's great. Super, thanks. So next slide, please. I've only got a couple of slides, and I think this is a short one. Basically, nobody really has a complete overview of the whole world of repurposing. There are some places that do it very well and have been doing it for a few years, but we can safely assume everybody around the table doesn't know everything. But what's important to know is what you don't know. If you want to do repurposing and you're at a specific point in the journey, whether it's the beginning or even near the end, what you don't know that's going to come up and sneak up and bite you in the butt is a major issue. So what's important is to be able to surround yourself with advisors, people like Phil, like Rick. There are many organizations that are out there to help you understand what's going to be, what are the hurdles going to be ahead of you, and who are the people that are going to try and actively stop you, actively help you, or just want to get reasonable evidence from you and want to know what is that evidence you want to generate. Because there are different pathways. There's basically a decision tree in repurposing. And you're going to start with a compound or a hypothesis or a patient, a disease. And then you need to know the drug that you're going to find, who does it belong to, who does the original data belong to, the data that shows that it's safe and efficacious in a previous indication, so you don't have to generate millions of euros worth of preclinical toxin safety data. Who manufactures it, who would like to manufacture for the new indication, who would like to go through the marketing authorization process in the future. Depending on the answers to those, you're entering on a specific route. And so you need to know which questions to ask, who to ask them to, and then how to engage the people that are on your specific route. Next slide, please. So basically, there are many organizations that can help. A lot are here today already. This workshop series is in fact organized by other organizations, but I'm going to name two because in the rare disease field, you come to either one of these and we're going to help you find these people, the other people that I haven't mentioned, and basically around the globe, we'll be able to find the people you need. So first is the European Joint Program in Rare Diseases. As researchers on the call, you'll probably be aware of this, but it's basically a very large public collaboration involving member states, funded partially by the European Commission and the member states, covering infrastructure and services and data solutions, clinical translational solutions and support for rare disease research and development. It can also be for new therapeutics, diagnostics, etc., etc. So any question related to rare diseases, you bring it to the European Joint Program, and you can go to the website, and there's a central help desk. Depending what the question is, it'll come to possibly EATRIS or Fondazione Telethon in work package 19 if it's related to repurposing. So that's the one address you need to remember. And if it's repurposing related, it'll find its way, and we will help you find the people that you need, including with the mentoring program in work package 19, where Anita and Agustin and the Fondazione Telethon team are expertly finding mentors from around the world to help you develop your drug development process, project, and repurposing project to the next steps. Secondly, another name that you should, this is a newly launched initiative called Newfound, and this is a collaboration between EATRIS, NIH NCATS in the US, as well the Cruz Foundation in Brazil, and the Open Source Pharma Foundation in India and US, who basically have come together around drug repurposing and trying to really create an open model of drug repurposing. And the first thing we're doing as we develop our competences to support the community is basically mentoring projects. So we're already actively mentoring projects, helping people ask the questions that they need to ask, understand the specificities of the project, and find the experts that can help them take the next steps. So these two addresses, any question you have, we will help you find the right people to help answer those questions. So turning those unknown unknowns into known unknowns, and then hopefully we get rid of them as unknowns at all. Next slide, please. In fact, there is no next slide. No, there is no next slide. So,
and there is
yes, sorry, yes, of course. So, very important organization to remember as well. And I don't know, I know Christine's on the call. So, Christine, do let us know. It can be any organization that approaches you, even though you're French-based, right? So the Fondation Maladies Rares, which has been around for about 11 years and have already supported 400 projects. And have over 150 publications and have really strong links between industry and academia and healthcare to really help with the rare disease development. Christine, please go ahead. I don't know if we can unmute Christine, sorry. Okay, no worries. Basically, you're going to hear. Yeah,
maybe. Hello? Yes, this is Daniel.
Hello, Daniel. Welcome.
Yes, I'm director of Foundation for Rare Disease. I don't know why. I know that Christine is listening, and I don't know what technical problem she has, but yes, we do. Actually, we are open to many international collaboration. We tend to call us Foundation for Rare Disease, FFRD, and French is only one part of our activity. And yes, we are open to submit proof of concept to our pharma industry partners, and they could come from other part of Europe, of course.
Outstanding, super, Daniel. Thank you. So this is also another address for you to remember. So there's one last slide on this, please, Rebecca. So the services at the foundation. So basically, the services that really help you to establish proof of concept. So it can also be repurposing, and there's also good links with industry. And I've heard stories from Christine before, and we're going to hear them in the next workshops of success stories, showing that engagement at the right time with the right people really can have a success. I think I heard Christine in the background.
Yeah. Thank you, Anton. Thank you very much. We have many projects at the French Foundation for Rare Diseases that we accompany regarding drug repurposing projects. And that's very interesting because 50% of these projects are clinician driven, which is quite normal, but also 50% are researcher driven. And among our statistics, we found that 1 project out of 5 is a clinical trial, but maybe we have a kind of bias at this stage, because the French Foundation for Rare Diseases has 75% of preclinical proof of concept. So for sure, sure there are more than that projects focusing on drug repurposing that are in clinical trials. Among our statistics, we also have 31% of cases that concern ultra-rare diseases. And in the vast majority of cases, small molecules are repurposed. And we always say that drug repurposing is not innovative, but it can be innovative. And we've got off-target drug repurposing in 20% of cases.
Outstanding, thank you. So indeed, you've got amazing experience there. So, and you are open and accessible. So do bear that in mind, folks. Right, okay, so I now hand to Rebecca, who's going to give us another poll, please. We have
just to kickstart the discussion and to learn more about the audience. Let me start the poll.
And then we're going to have a good 20 minutes of discussion. So feel free to throw more questions into the Q&A, and we'll also open the possibility for people to speak.
So it's actually 2 questions for you to answer. Yeah, I see questions, answers coming in. I will give it a little bit more time because answers are still coming in before I close the poll.
Interested to hear on the second question, the compound you're using, what other could be? Could you maybe, if somebody who has answered other in the second question in the chat, if you could explain what the nature of the drug is, if it's not on the market and generic, that sort of thing, that would be interesting to hear, please. Very interesting to see that as quite a broad, so that we have all sorts of examples, and each of these is a very different type of strategy as well, I believe. So depending on whether it's on patent, off patent, whether it's been actively manufactured or not. So that shows some of the challenges we face, is that there's no one single answer. Nice to see that so many of the audience are actually actively involved in repurposing right now, but also there's still a lot of interest as well of people that don't. Super. Rebecca, anything further on this?
No, not from my side.
Yeah. Somebody named Tiago has mentioned doing pediatric formulations. Now that's a very interesting one. It's like, in a way, it's repurposing, or it's extending the purpose, in the indication, but outside into a different age group. I think that's a very interesting element, and personally I would consider that a very important element of the repurposing strategy to ensure that pediatric populations are being tested. Thanks for sharing your experience there. Now, thanks, Rebecca. I think we can leave it there. I invite Phil and Rick to turn on the cameras again. And we also are happy to hear further questions from you. If there was anything that we missed earlier as well. I had a question actually to Phil. You talked about the signature mapping, and you said there's a lot of interest in there right now. Have we seen yet any successes of programs that have reached fruition based on signature mapping?
Yeah, so I couldn't find one, but I know that we've been trying to include it in some programs internally, but I think as a sort of a standalone effort, it might be quite tricky to get it to work, because bringing your own experimental data in there, typically you might just be relying on external data resources to do that. And it's the same with the connectivity mapping. So I think there's a sort of a dependence on the quality of these external resources, and typically they're pretty good, yeah, but it isn't necessary. It may not reflect the nuances of your disease or your patient population or the tissue that you're interested in sampling or your in vitro model. And ultimately you have to come up with a proposal for a compound and test it. So I think when I was getting ready for this, I did have a look through it, and we have included sort of this reverse genetics story, but it's not straightforward, and it doesn't give, there's no clear answers, unfortunately.
So I'll chip in on that one as well, Anton. It's fascinating, and there's lots of companies I know that are playing about with this kind of technology and fusing that reverse genetic stuff Phil was talking about with a range of other AI approaches and literature mining to try and delve into the potential repurposing candidates. And I think, firstly, the point they always make is what Phil's just made, that the data sets that they're using are absolutely crucial, and they find a lot of the data doesn't reach the standard that they necessarily need to get really robust information from it. Secondly, that they're always feeding in supplementary information on top of that signature mapping to make sure it's not the only thing they're basing their hypotheses on, and that includes a kind of human expert kind of review of the suggestions that come out of there to really pick on the things they think they can see the strongest logical basis for, which is interesting. I know of a few projects through those kind of companies that have got to in vivo or in vitro testing and have found positive results of the hypotheses, which is great, but I've not yet heard much about anything that's gone beyond there and led to a kind of clinical testing. So I think it's really exciting science, and it sounds fascinating and amazing when you see cases where they've gone from a rare condition that's got no hypotheses to a tested in vivo success within a matter of months. That's really rapid, but how things then move to the next stage beyond there, I think, is still a fair way to go in the experience I've seen.
Clear, thanks. This is a question to you, Rick, for the academic researcher out there who maybe through computational methods and pathway analyses has stumbled upon a potential for a new disease for a given drug, but hasn't actually engaged the patient community yet. At what stage and how would that academic engage a community?
That's a really good question, actually. I don't think it's ever a wrong stage. If you've got any kind of interest whatsoever in a rare condition, I think you're going to gain quite a lot of valuable insight very early on from reaching out to a patient community if it's out there. So how is probably more the question. I think any kind of online search you can do to delve into groups and organizations that are out there representing that disease or related diseases is probably your best bet. You could probably have a look at some of the kind of umbrella organizations. EURORDIS at the European level will have a list of a lot of fully functional registered patient organizations in rare conditions, but there are many national bodies as well. So we have Rare Disease UK in England, there's obviously NORD in the US. A whole range of those will list a number of charities that be very open to engagement, and nearly all of those will have very simple contact forms to reach out, and they'd be thrilled to be contacted by researchers. Many of them spend majority of their time trying to send out emails and knock down their doors. And I think if you then make that contact, the key thing from my perspective is to be open, clear, and not elevate expectations too high initially. Say what you found. Say why you're reaching out, and say what you think you can do in a fair way. Don't overpromise early and vanish, because that's going to break trust straight away. And building trust with those relationships is key, because if you can do that, these communities have access to data, whether it's anecdotal, through interviews, or through detailed registries that can tell you a huge amount about the lived experience and actual progress of the condition, which can help you take that concept to a place where you can actually execute it in a much more effective manner.
Yeah, very good advice indeed. That the expectation management of expectations of patients, especially in such acute diseases as often is the case with rare diseases, is very important. And I think there are a lot of examples of that over-promising and under-delivering from the research side. Thanks, Rick. The question in the Q&A box, I think is a good one for you, Phil, regarding polypharmacology and the synergistic effect for a single disease. Do you think it's possible to follow the same pipeline, or will the regulator want a bit more scrutiny because it's doubling up?
Yeah, so I'm not the expert on this, but I imagine that there will be some more. If this is something that is, say, combined in a single tablet, or in a or where the effective one is synergistic with another, and I think it becomes very important there to understand the sort of dose relationship with each of them, and both of them together, and what impact that might have on toxicology as well, and on safety. So I'm not aware of too many situations. Obviously, it's happening a lot now in the sort of COVID-19 area, where people are actually running in vivo studies looking for synergism between remdesivir and compound X that they've found. And also there's a combination of drugs of Desivir and Veklury or something, which is another marketed drug, which is being used in a repurposed setting. But I would imagine as soon as you get into sort of where synergism is important, then those kind of synergism effects can happen over quite sort of narrow concentration ranges, or they might be very dependent on the metabolism of one of the components, which might be happening at a more rapid rate than the other component, and the ADME, PK, and PD properties might be very different in different populations. So I would just be careful, and it's one of those ones where you need to check with the regulator. But what we do see though is a lot of the compounds which come up in our repurposing screens are quite noxious compounds, sort of old school anti-cancer compounds always seem to come up with an alarming regularity, mainly because they're sort of binding to DNA and other sort of very non-specific effects affecting cell growth and metabolism. So if you do have a synergistic effect, that can mean that you can reduce the dose of one of the compounds which potentially is more toxic, then that is a good thing. But again, you need to I think we'd have to square that with the regulator. So I'm all for it, but it does make life a lot more complicated, and there has to be a good reason behind it.
Yeah. Great answer, Phil. Thank you. And I saw that Pan Patriarca from the Anti-Cancer Fund has raised his hand. Pan, I'm sure you have something very helpful to say on this. Please go ahead. I assume that audience can unmute themselves.
Not yet. Yes, I need to just do it. Give me time to find the right.
Yeah, thanks.
Right window. But there's another question in the chat. But maybe we start with that one, and then I.
Okay, that's a good idea. Yeah, sure. So this one I could maybe have a go at, but also Rick, I'm sure you know a lot of this. So what's the current situation with financing? Who's financing repurposing? And I think it very much depends on what the opportunity is. If it's an opportunity where somebody could invest the money and make money because there's intellectual property protection they could get, or they could extend the intellectual property protection of their existing drug, then people will be very glad to finance it, and you're not going to have issues there. That's the dream scenario, except you probably end up with a high price drug. But at the end of the day, that's a different problem, which is basically drug pricing. But there's a lot of cases where there's a generic drug for the indication sold at just a few cents a pill, and that's always going to be there. So who's going to invest money to go onto a market where the same drug is being sold for a few cents a pill? And that is not going to get the at-risk financing. So you're going to have to then look at European Commission, charities, philanthropies who are going to want to take that journey with you. Rick, anything to add to that?
You pretty much covered it very well. I think just to add that generally the larger pharma we're seeing don't want to take any kind of projects like this on until you've got some clinical evidence of efficacy. And before that, they're very risk averse. But when you've got that, there can be a much more positive move. And there are a number of SMEs that are moving some repurposing opportunities where they think they've got a route to market through reformulation at an earlier stage, and that they are happy to play in the ultra rare space as well. But generally speaking, yes, if it's ultra rare and generic, you're going to be scraping around for back of the sofa change often.
But there is increasing commercial interest also from venture capital. I know at least one that's rare disease specific venture capital fund, but not necessarily for repurposing, but rare disease specific. And I think if they could see a return on a repurposing trajectory, they would be equally open to that. Thanks, Rick. So Pan, your mic should be open now.
Yeah, just a comment following on from what Phil said about pharmacology, just based on our experience at the Anti-Cancer Fund, where we've suggested a generic drug in combination with chemotherapy. What was very important was for the regulator was showing whether there was any single agent activity in the repurposed drug. So even though we weren't proposing the repurposed drug as a monotherapy, they wanted to know what was the effect. And so that's when you have to start thinking of things like window of opportunity trials, where you can give the drug as a single agent before some other treatment, just to show, A, that the drug is safe, and is there an effect independent of the synergy.
Nice one, Pan. Very good advice and feedback. Thanks very much. Much appreciated. I have one question to Phil, yourself. So in the drug screening process, and there are these great libraries, right, ranging from 3,000 to 8,000 compounds. what's the typical hit rate? Are you going to get 5 compounds that you have to check, or is it going to be 50? And how do you quickly whittle that down, without spending insane amounts of money to some interesting leads?
Yeah, so that's a good question. So on the library size, just on that library size point, everybody thinks that their library is the perfect size, and it doesn't matter how big the library is. So you go to a pharma company with 1 million compounds, and they say that's the minimum. If you go to somebody who's only got 1,000 compounds, they say that's more than enough. So there's a degree of subjectivism about the whole question. I think when it comes to sort of hit prioritization and hit selection, so you always get hits. So typically the hit rates are about 5-fold higher than you would get with a library of diverse chemical structures. Yeah, so you're always getting more hits, especially in cell-based assays, because these are pharmacologically active compounds, and many of them have got dozens of secondary activities which are described in literature, plus other activities which are not yet described in literature that can be only revealed in your assay system. So I think what you have to really try and understand is, or what you have to have a good handle on, are the sort of enriched data and metadata that come along with these compounds. So I think the best resource that I've seen is the one provided by the Broad Institute, which supports this collection that they assemble. So this CLUE-IO database. So not only is it able to hold information about the biological activities in target-based assays or antibacterial effects or antifungal effects, it's also got all of these data which are looking at changes in expression, changes in the transcriptome, the effects in terms of the cytotoxicity against a wide range of different cell lines. So you've got all of this really interesting data. Now, it's not perfectly aligned to your program, so you have to tune it to that, but that will give you a sense of what the underlying targets may be. And then you can use that information along with other information. So they have practical information there about what is the patent status of the compound. So if you're, if you're collaborating with Merck on corona anti-COVID drugs, which is in this molnupiravir that was found out by some guys in California doing a repurposing screen. If you can collaborate with them, then you can advance that drug. Yeah, because you have that collaboration. But if you're only able to follow up using the generic hits which are in there, then you can get a hold of those. So it's all about using the data and using the annotation, and then sort of having a rational approach. And that's when it's actually quite handy to have an experienced medicinal chemist on your team. So we're not doing new structures or structure activity relationships, but a really experienced medicinal chemist who's been around the block, because compounds in the clinic, they can really look at these hit lists and give you a real insight into it. And if you get 2 medicinal chemists, then you'll get 3 different opinions about what you should advance. But I mean, having those people is, I think, essential. Having that sort of expertise, and typically coming from a background in the pharma industry. So I would say, yeah, it comes down to judgment and data, but you need both things as well.
Yeah, thanks. I think that's a really nice closing remark, because what you're implying there is that this is a phenomenally multidisciplinary undertaking that goes way beyond the laboratory and the multidisciplinarity of the science itself, which is vast. But then we also have to engage the clinician, the regulator, the health technology agency, the patient, and have the patient driving these forward, because you're not going to take it all the way as a scientist. So it's, I think, nice to see that there's so much interest in repurposing, but it is early days yet as an industry, as a discipline. So there will be policy work over the next years, but I think what's important is that we as a community come together and really look at what these issues are, try to help each other with expertise like Rick's, like yours, Phil, to help people understand what are the challenges you're going to do, and that's to show that it's also not all so rosy. There are so many confounding factors, both in the science and in the market itself, that it's an incredibly difficult undertaking. I think we're going to wrap it up here. I think Rebecca has one last slide for us to remind us of some very important things. predominantly that next week, same time, we have another session. So today was very much a general introduction with this really insightful and excellent presentation from Rick and Phil. Thank you for those. We're very grateful for taking the time. Next week, we'll go a little bit deeper into the challenges of actually transversing that pathway towards the clinic around regulatory issues and that. And also then in the following week, we'll look more around the market-based issues. So thank you all very much for your attendance today. We hope to see you again next week. Do invite your colleagues. See in the chat, there's a link there to mention to other people, because the more people that know about this, the more of a community we can create and the more opportunities there are for sharing with your colleagues. So huge thank you to the organizers, to the speakers, and to yourself, the audience, for joining today. Looking forward to next week's session. Thanks again.